Hello, my name is Michelle Pelchat, and I'll be discussing Unit 2 case studies. The first case that I chose was Case 1, which discusses pyruvate kinase. Part 1 question is, under normal conditions, describe all the ways in which pyruvate kinase is regulated in the cell. So under normal conditions, there we go. There are multiple ways which pyruvate kinase is regulated in the cell. One of the ways is by phosphorylation, while the other way is by allosteric means. So phosphorylation is regulated by the glucan-mediated phosphorylation, as noted in the top part of this diagram here. So in the fed state, when the glucose levels are high, pyruvate kinase is then dephosphorylated and considered active. In the fasted state, glucagon levels are elevated, which causes a cascade of events leading to the phosphorylation and inactivation of the pyruvate kinase, as seen on the right side. It's also regulated by allosteric means, as noted on the bottom, by feed-forward activation by fructose 1,6-bisphosphate, which allosterically activates the enzyme, as noted on the bottom, and increases the flux through glycolysis. You can also see here that ATP allosterically inhibits pyruvate kinase in order to slow glycolysis when the energy state is already high. Part 2 of the question discusses more of the cancer cells and how cancer cells predominantly express type M2 pyruvate kinase as opposed to the other three. So these are the 4 types of isoenzymes of pyruvate kinase. As you'll see here, PKL in the hepatocytes mainly, PKR in the red blood cells, PKM1 normally in the heart, skeletal muscle, smooth muscle, and brain. PKM2, as you'll see here, mainly found in the tumor cells and of course in embryonic tissue. So these are the 4 isoenzymes of pyruvate kinase that exist in all mammals. Cancer of different tissues have been associated with an increase of the M2 isoform, as seen here. Type M2 is characteristically found in fetal tissues and progressively replaced with one of the 3 isoforms during normal fetal development. M2 is regarded as allosterically regulated and due to the sigmoidal kinetic response to the binding of the PEP. It also is inhibited by ATP. PEP is converted to pyruvate via pyruvate kinase during glycolysis, as noted earlier. So malignant cells have a high rate of glycolysis, and it has been stated that the shift towards the M2 in the tumors represents a mechanism to gain allosteric control of the enzyme and allows for activation and inhibition of the enzyme during the cell cycle. PKM2 enzyme activity is subject to complex allosteric regulation, which is crucial for tumor progression and growth. It can be found in a tetramer form, as noted on the right side here, which has a relatively high activity, in dimer form, which is low activity, which is what you'll see for the cancer cells. Has low catalytic activity, resulting in the increased anabolic synthesis of macromolecules through the pentose phosphate pathway, also known as PPP, which then promotes cancer cell proliferation and growth via production of glycolytic intermediates, such as fructose 6-phosphate and glyceride 3-phosphate. The PPP also results in producing NADPH, which is high-energy compound used for reductive biosynthesis because it cannot be oxidized in the electron transport chain, and it suppresses the reactive oxygen species production. NADPH is needed for nucleotide biosynthesis, which nucleotides are then needed to support the rapid cell growth. The dimer PKM2 reduces catalytic activity and participates in macromolecule biosynthesis, which then generates the materials needed for the rapid growth that you see in tumors. In the dimer form, PKM2 can also serve as a cytosolic receptor for thyroid hormone and is involved in gene transcription, where it enters into the nucleus after oncogenic stimulation, where it acts as a protein kinase and phosphorylates proteins, including histones. An upstream glycolytic intermediate activator of PKM2 is called fructose 2,3-bisphosphate and is involved in the allosteric regulation of the dimer form. So in the next slide, you can see the difference between cancer cells and normal cells. Cancer cells are often identified by their reprogrammed pathways shown here, which can be considered the Warburg effect, or anaerobic glycolysis. Cancer cells will take up glucose and then produce lactate regardless of the oxygen availability, which you'll see through here, which invades metastasis and creates less ATP compared to anaerobic glycolysis. This increase of glycolytic intermediates supply the subsidiary pathways to complete the metabolic demands of the quickly growing cell, as seen on the left side of the cancer cell. PPP does nucleic acid synthesis. You also have down here acetyl-CoA going into fatty acid synthesis. The two of those together really support that cancer cell growth, which is why they choose to do M2 as their isoform, because they can rapidly grow more so than the average cell. The other case that I chose was case 4, which discusses a healthy 2-month-old female that presented with jittery spells several hours after breastfeeding. Her mother reports she had not given the baby anything to eat other than breast milk since birth. Baby has only recently been sleeping for more than 2 hours between feedings, and that's when the mother reports the symptoms started. Laboratory results are consistent with hypoglycemia, which is low blood glucose, but are otherwise unremarkable. Epinephrine is administered and there was no increase in blood glucose, but there's an increase in blood lactate with the epinephrine. Physical exam reveals liver edge 4 centimeters below the right. Costal margin, percussion of the right chest and abdomen confirms enlarged liver. Blood glucose levels increase after breastfeeding, but she can't maintain the glucose within a normal range during fasting. So that's when you're seeing the hypoglycemia. So I've bolded some of the things that are important. First thing being breast milk, which is considered lactose. Lactose is made up of glucose and galactose, which means that we're dealing with these two substrates, not fructose. That'll be important in ruling out option A. The other thing that I did bold was the epinephrine that was administered. Epinephrine isn't present during the fasted state and is responsible for catabolic pathways. Different pathways activated by the hormone include glycogenolysis and glucagon-mediated lipolysis. So since the epinephrine caused no increase in blood glucose, but there was an increase of blood lactate, and lactate is produced when glycolysis enters the anaerobic pathway. So the other thing to notice is the symptoms that they stated. So the large liver, poor regulation of blood glucose in the fasted state only, and no positive effects of epinephrine, and the fact that we're dealing with lactose, not fructose. So part A, automatically, or option A, the hereditary fructose intolerance, dealing with fructose, and the metabolism of fructose, which breast milk does not contain fructose. So that's been eliminated. Even though some of the symptoms are similar, such as the enlarged liver, still we're not dealing with fructose. So we can already rule out option A. Option B is galactosemia, which is caused by a deficiency in galactose 1-phosphate uridyl transferase, which is known as GALT, during galactose metabolism. Galactose is typically ingested as lactose. The galactose is taken up by the cell, phosphorylated into galactose 1-phosphate by galactokinase. Galactose 1-phosphate is then activated to a UPD sugar by the GALT. So a deficiency in the GALT would block the galactose metabolism in the liver and would lead to an accumulation of galactose 1-phosphate, which would inhibit the hepatic glycogen metabolism, along with other pathways that require the UPD sugars that aren't being generated. So that's toxic to the kidneys and the central nervous system and can be fatal, but that wouldn't increase blood lactate levels, but not the blood sugar when given epinephrine. So that's ruled out because that would have no effect. And you can see here that epinephrine was administered and there was no increase in blood glucose, but this would have that increase. And this is showing that there was an increase in blood lactate, which GALT has no effects on the blood lactate levels. I'm going to skip C and go to D, glucose 6-phosphate dehydrogenase deficiency. That's a key regulatory enzyme of the pentose phosphate pathway that I was talking about earlier during the irreversible oxidative phase. So that would cause a decrease in NADPH production. It would present as a hemolytic anemia. It would demonstrate jaundice. Anemia was not discussed during this child's symptoms, nor was jaundice. And this would also have no effect on the lactate levels. Therefore, that is also not the correct answer. So that leaves us with option C, which is glycogen storage disease. In week 6, we learned this as the von Gerk's disease. So the lacking enzyme in von Gerk's disease is glucose 6-phosphatase, which is required to dephosphorylate glucose 6-phosphate to then generate free glucose, which is seen at the top of this diagram here. If we're not able to generate free glucose due to not having the enzyme of glucose 6-phosphatase, then that would impact gluconeogenesis and glycogenolysis processes and would lead to a fasting hypoglycemia due to many other pathways being involved here, as you can see, which were some of the symptoms that the child displayed. So when glucose 6-phosphate is lacking, the glucose cannot be released from the liver and into the bloodstream to normalize blood glucose levels, which would then lead to the fasting hypoglycemia on the top. The increased glucose 6-phosphate levels would also lead to increased pentose phosphate pathway, along with increased glycolysis to pyruvate and glycerol 3-phosphate. Increased levels of pyruvate in the cell leads to 3 further pathways, including increased lactate production via lactase dehydrogenase, which is LDH in the picture, increased alanine via alanine transaminate, ALT in this picture, and increased production of acetyl-CoA via pyruvate dehydrogenase complex, PDHC in this picture, showing on the arrow towards the right. Increased levels of glycerol 3-phosphate via glycolysis and the glycerol 3-phosphate dehydrogenase, which is GPD1 in this picture, are then synthesized with fatty acids, leading to increased triglycerides, which will then lead to the hyperlipidemia, as well as fatty infiltrations causing the enlarged liver. That's all shown on the right side of this picture. So von Gerk's most best out of those 4 options describes what that child has for a disease based off of the hypoglycemia in the fasted state, enlarged liver, along with the increased lactate when given epinephrine. So the correct answer here is D. And that is all. Thank you.